The complement system. Soon after the discovery of antibody by von Behring and Kitsato in Berlin in 1892, Bourdieu and Weber discovered another factor in animal sera that complete the effect of antibody on antigen molecule. Complete the effect of antibodies on antigen molecule. This factor was termed elixir, but because of the complementary effect on the function of the antibody, Ehrlich called it complement. There are several different complement components forming that is called the complement system. The components of this system are capable of specific sequential interaction leading to myriad physiological and immunological effects. The general properties. The complement system consists of complex series of interacting proteins that are found free in body fluid or bound to cell membrane. Some of the complement components are designed by the uppercase letter C followed by the number of C1, C2, C3, which is related more to chronology of its discovery. Other complement components are designed with the letter of alphabet factor B, H, I, D, so forth. Complement components constitute 15% from the total serum protein. They are globulin in nature and most of them are beta globulin. Few of them are alpha 2 or gamma globulin in nature. Its molecular weight range from 40 to 40 kilodalton. Serum concentration of complement components range from high as 120 mg per deciliter C3 to virtually undetectable level like C2 or C9 complement component are heat labile and become inactivated at 56 centigrade for 30 minutes. The complement activities are not species specific and its complete non-specific, the immunological specific effect of antibody on antigen. Complement components are found in body fluid in inactivated form and their activation occur in sequential manner. Activation of one component lead to activation of the second one and so on. The most important event in complement activation is the cleavage and activation of C3 component. The protein of complement system from two interrelated pathways for cleavage and activation of C3, which is the central event of complement activation. The process of complement activation is associated with release of low molecular weight peptides that have numerous biological activities. These peptides are identified with lowercase letter as suffix C3b. Activation of the complement system has three main effects that include cell lysis, cell activation, and enhancing of phagocytosis. Discovery in 1894 by Bourdieu. It represent lytic activity of fresh serum. Represent the lytic activity of fresh serum. Its lytic activity destroys when heated at 56 for 30 minutes. The host benefit from the complement function, opsonization to enhance phagocytosis, phagocyte attraction and activation, lysis of bacteria and infected cells, regulation of the antibody response, clearance of the immune response, clearance of apoptotic cells, inflammation, anaphylaxis. C. Activation, alteration of C protein such as they interact with the next component. C. Fixation, utilization of C by antigen antibody complex, hemolytic units, dilution of serum with lyse 50% antibody coated in as a suspension. C. Activation, the nature usually by heat of an early C component resulting in loss of hemolytic activity. convertase esterase altered C protein, which act as a proteolytic enzyme for another C component. Proteins of the complement system nomenclature: C1, C2, C3, C4 by number, factor B1, B, B, D, H, B, D, H, and I, protein B, mannose binding lactin, MB associated serine protease, C1 inhibitor, C1 INH serbin, C4 binding protein, decay accelerating factor, C1 receptor, protein S, vitronectin. Activated components are usually overlined like this. When enzymatically cleaved, the larger moiety binds to activation complex or membrane, and the smaller peptide is released in microenvironment. Letter B is usually added to the larger membrane binding peptide and A to the smaller peptide. Except C2, the larger membrane binding moiety is C3, C2A, the smaller one is C2B. Classical pathways of complement activation: classical pathway, lectin pathway, and alternative pathway. Classical pathway, antibody dependent; lectin pathway and alternative pathway, antibody independent. Activation of C3 and generating of C5 convertase. Activation of the C5 lytic attack pathway. Component of the classical pathway: generation of T3 convertase, C4 formation, C4b, C2, C2a, C2b, C4b to A is C3 convertase, C3 added, C3a, C4b to A, 3b is 5 convertase. It leads onto the membrane attack pathway. Mannose binding lactin pathway, MBL, MAB1, MASB2, C4, C4A, C2, C4B, C2B, C2A, C4B to A is C3 convertase, will lead to the generation of C5 convertase. Component of the alternative pathway: C3, H2O, generation of C3 convertase, B, D, BB. C3 IBB complex has a very short half-life. C3B, C3 and C3B. C3 activation, the amplification loop. If spontaneously generated, C3B is not generated. C3B, C3BB, 3BB. C3 activation, the amplification loop. 3,3B formed BB, C3B, C3A, added B, D, C3B. Control of spontaneous C3 activation via DAF prevent the binding of factor B to T3B, C3B. DAF dislodge T3B bound factor BB. It's a cascade of events. C3 B. Bind and activator protector membrane. C3b stabilization and CC5 activation. This is a stable C5 convertase of the alternative pathway. C3 regulation on self and activator surfaces, autologous surface and activator surface. C5 convertase of the two pathway. C5 convertase of the classical and lectin pathway. C5 convertase of the alternative pathway. Lytic pathway. Generation of C5 convertase lead to the activation of the lytic pathway. Component of the lytic pathway C5, T8, C6, C9, C7. Lytic pathway. A symbol of the lytic complex. And here CQRS breakdown. C1 inhibitor deficiency, angioderma. Opsonization and phagocytosis. Opsonization, binding, and phagocytosis. C3b, C3b, IC3b, C34. Biological properties of C activation. C2b, bronchoalinin. Biological effect, edema. C3a, anaphylaxis. Mast cell degranulation, enhanced vascular permeability anaphylaxis. Carboxyl peptidase. C3INA. C3b opsonization, phagocyte activation, regulation, factor H and I. C4a anaphylaxitoxin, anaphylaxitoxin, C3 but less potent. C3INA. C4 opsonization, phagocytes, the regulation. C4 4b factor 1. C5 chemotactic factor, biological effect, anaphylactic as C3 but much more potent, attract activating BMA, cause neutrophil aggregation, stimulation of oxidative metabolism and leukotriene. C5b6-7 chemotactic, attached to other membrane protein